is what we are focusing on. Here's the location of the electron transport chain. Here's the mitochondria. Here's the matrix, which is the inner portion of the mitochondria. Here is the membrane. We have an outer membrane, and then we have an inner membrane. And so the location of the electron transport chain is in the inner mitochondrial membrane. If we were to zoom in on that and actually look at what's happening, these are the molecules, the complexes that are inserted into the inner mitochondrial membrane. And then at the very end, this is ATP synthase. This is the most important complex that is going to help us generate ATP. So the electrons that we've shuttled here, we can see them here, they're going to be passed from our carriers into complex 1. You can see them going from their reduced form back to their oxidized form as they pass those electrons over to complex 1. Now, every time we pass an electron from one complex to the next, we move some of these hydrogen ions from the matrix into the intermembrane space. So when we pass an electron from complex 1 to complex 3, we actually use that energy to move these hydrogen ions against their concentration gradient. Remember, when we have a concentration gradient, molecules tend to want to move passively down that gradient from a place of high concentration to a place of low concentration. So in order for us to move molecules against their concentration gradient from a place of low concentration to a place of high concentration, we're going to need to yield that from energy. So that is what the electrons are doing. So as we pass them along the chain, we're generating a very strong hydrogen ion gradient. At the very end of the chain, we're going to pass the electrons from complex 4 down here onto oxygen, right? Remember we said that oxygen is the final electron acceptor. This is the whole reason that we breathe oxygen, to go into our mitochondria, to stand in the matrix, and to accept these electrons as they're passing along the chain. When that is done, we can now convert oxygen into water as a byproduct. And then we have now generated this really fascinating gradient. Now we have lots of hydrogen ions in the intermembrane space, a few hydrogen ions down here in the matrix. And so now we can move these ions passively down their gradient through ATP synthase, and that is how we generate ATP. So we have about 34 ADP here standing by, waiting to be phosphorylated. And the energy to do that is by moving the hydrogen ions down ATP synthase and taking that energy and phosphorylating 34 ADP to make 34 ATP via the electron transport chain. Okay, so that is the final stage of cellular respiration. And we need oxygen for this stage. That's, again, the whole reason that we breathe oxygen. Its sole purpose is to be a part of the electron transport chain, oxidative phosphorylation, and to generate a significant amount of the ATP we need to keep us alive, to sustain our processes, etc. So that's kind of an overview, again, because this can be a little bit difficult to understand. I'm going to go through this one more time in a bit more detail, and then I'll see what questions come up at the end. So here is our intermembrane space, our matrix. Here's the inner mitochondrial membrane. And we can just, for fun here, we can also see the structure of this membrane should be very familiar to us, right? These are phospholipids. And just like the phospholipids that form our cellular membrane, all the membranes in our cells, even around our organelles, are also formed by these phospholipids, right? This phospholipid bilayer. Now, we have our complexes inserted in this membrane as well. And then we have our gradient that's going to be generated. The first thing we want to think about is the electrons that we have been collecting through glycolysis, through the linking step, through the Krebs cycle. We're going to bring them all here into the matrix, and we're going to pass them off to complex 1. That is going to return our carriers back into their oxidized form. Now we're going to pass those electrons from complex 1 to complex 3. When we do that, we move some hydrogen ions against their gradient. That's the reason these arrows are red. It's indicating to us that this needs energy. This is active transport moving against the natural gradient. And so the energy that we use to move these hydrogen ions comes from the passing of the electrons. Again, we pass from complex 3 to complex 4, and we generate some more hydrogen ion movement, which is strengthening our gradient here. Now we have a much higher concentration of ions in the intermembrane space and a lower concentration in the matrix. At the very end of our chain, oxygen is going to accept those electrons as the last stage here, and it is going to use those electrons and convert that into water as the byproduct. But the most important thing has already happened. We've generated a very strong gradient, a high concentration of ions. And so the final step of ATP synthesis is now to pass those electrons, or not electrons here, but hydrogen ions, down this ATP synthase. And now we're going to make ATP by phosphorylating ADP. Okay. So that is the synthesis of ATP. And I want to zoom out here and look at the entire cellular respiration again once more. So we had glycolysis, small amount of ATP, 2 net ATP. We also generated some electrons here. Then we had the linking step, generated more electrons, some CO2, but no ATP. This is part of the reason why some literature does not consider the linking step as an actual stage, just because there's no ATP generated in this second step. The third stage here is the Krebs cycle, more electrons generated, more CO2 generated, and then 2 ATP. So far, we've generated 4 net ATP before we even get to oxidative phosphorylation in the mitochondria. And then the final step here, we have oxidative phosphorylation, which is going to generate, again, anywhere from 28 to 34. Our number is going to be 34 of the total 38 ATP. Okay. All right. So here's the overall reaction. We had 1 molecule of glucose to start. We inserted oxygen. Again, this is an oxidative process, especially the very last stage. And then we phosphorylate 32 to 38 ADP, and we generate 32 to 38 ATP, okay? All right. Now I want to talk here a little bit about what happens to glucose in the absence of oxygen. Now, if there's low oxygen availability, let's say you're at a high altitude, for example, or you're just not generating enough oxygen or bringing enough oxygen, your respiration rate is low, what's going to happen is the electron transport chain is going to back up. Because we don't have oxygen to accept the electrons, the chain of passing those electrons from one to the other halts. Okay, that chain stops. Essentially, ATP production stops. All of the preceding steps will also stop. The Krebs cycle will stop moving. Glycolysis will continue to generate NAD, but that is because of another enzyme that's involved, lactate dehydrogenase, okay? I just want to point that out here. So we can still generate ATP through glycolysis because lactate dehydrogenase can take that pyruvate and make lactate out of it as sort of a byproduct instead of shuttling it off to the Krebs cycle. So this is why if we are, for example, sprinting, we're running a sprint, our rate of breathing is slowed or it's not enough to keep up with the demands of our muscle, We generate lactate because we are generating ATP anaerobically. Our oxygen supply is not enough to match the amount of ATP that we need to power our muscles for those contractions. So as a result, we shuttle that pyruvate off to lactate, and this is the lactate that that actually is shuttled to become lactic acid. This is why we feel that burn in our muscles whenever we're sprinting or any sort of activity or exercise where our respiration is not matching our metabolic demands in terms of ATP production. So this is sort of a backdoor pathway to be able to use this pyruvate and turn it into something else to keep ATP production going. This is how we generate it anaerobically.